representing sperm and egg up here. Because he's a sperm, but she's forlorn. She's waiting for the sperm to arrive. Just a little. I know.
Sperm have friends, eggs are alone.
All right, so now we need to do this part. So we're going to make a baby. So what do we call this process where the happy sperm and the forlorn egg come together?
Fertilization.
Conception would be one answer. You can say that. That's fine. The other one you could use is?
Fertilization.
Fertilization is another word. They're both fine. So either one's fine. So what have you made when you do that? So let's draw that on there. This is not anatomically correct. All right, so what do we call that product of conception?
Fetus.
No.
Oh.
No. Zygote.
Oh yeah. Oh boy.
A la Biology 112. You can also call it a fertilized egg. Those are both synonyms. So there's two words for the process, two words for the thing. I will talk about green one. Where's that? All right, so we have now put the sperm and the egg together. In biology, the land I live in, we call this time zero. We start a clock at conception. Doctors do a different thing. They're in their own world. But in biology, this is where you're going to start the clock and start timing events as we go through gestation. So for this class, this is 0. Right? So what are we going to do now that we have a fertilized egg? What will happen next? What will it do first?
Divide.
It will divide. So step number 1 after that, we call it cleavage. In this context, or in any context, what does cleavage mean? To divide. Right? So the zygote will divide. Start dividing. If you remember your biology 112, what's that M word for dividing?
Mitosis.
Mitosis. So we can also call this mitosis. Not meiosis, because we already made the sex cells. So we're going to undergo mitosis, or cleavage, and divide. So where's it going to divide? In the fallopian tube, or uterine tube, or oviduct, pick the word you like. So we're going to divide the fertilized egg, the zygote, into more cells. I'm going to show you a picture of that, and we'll add some detail. So, in this picture, ovulation, there's my eggs coming out. Uh-oh, guys with tails. All right, so congratulations. Here's your zygote in the fallopian tube. Becomes 2 cells, becomes 4 cells, on down becomes a raspberry, right? Becomes a big tennis ball. So what I'm doing is I'm dividing or cleaving this zygote into different pieces. What am I going to do after I do cleavage? What's next? Implantation. So there it is. We're going to implant. So step 2 is implantation. Implant where?
Uterus.
It's the uterus. It's the endometrium. You learned last week and was on your test. So we're going to implant into the endometrium. Now we got to come up with a timing. So if time 0 was zygote, how old are you in that picture? What time is implantation? All right, what? I heard something.
7 days.
Or 6 days, 7 days.
1 week.
1 week. 1 week is usually the answer we want. So this is week number 1. At 7 days, roughly, after conception, you found mom's uterus. Right? So you have now implanted week 1. It's called the rule of weeks. So week 1 to get here. Okay? So I'm going to show you an animation of that process of 1 week. Yeah. Boom. Put that off for a second.
Fertilization typically takes place in the upper part of the oviduct.
Fallopian tube.
Cleavage begins as the zygote moves through the oviduct toward the uterus. Continued mitotic divisions produce a ball of 16 to 32 cells called a morula. By the 5th day, a blastula has formed, with a surface layer of cells surrounding a fluid-filled blastocele and an inner cell mass.
Okay, so in case you read Saladin, which I don't, you will never find the word blastula anywhere in that book, because blastula is a word used for everything but humans. So if you're looking up cows, sharks, sea urchins, chickens, they use the word blastula. to mean this tennis ball. You are called a blastocyst because we're special because we're people. So blastocyst and a blastula are the same thing. There's no difference in what they are, just different words. So humans, we use a different word. So remind me from 231 what blast means. Create, form, immature. So blast you something means an immature person, immature thing. So your blastocysts, blastulas are just meaning they're immature. So don't freak out if you see blastula some places and blastocysts somewhere else. This guy's using a non-human word with a human picture. That's fine. All right, so don't freak out on that word.
About a week after fertilization, implantation is underway. The blastocyst adheres to the endometrium that lines the uterus and begins to send out projections into the maternal tissues.
Yeah.
As implantation proceeds, the inner cell mass develops into an embryonic disc that is 2 cell layers thick. This will give rise to the embryo. Membranes start to form around the embryonic disc. The amniotic cavity will fill with fluid and cradle the embryo. The yolk sac will function in blood cell formation. Spaces in the maternal tissue around the implanting blastocyst open and fill with blood. Inside the blastocyst, a chorionic cavity opens around the amnion and yolk sac. The membrane that lines this cavity is the chorion. It will become part of the placenta. Now let's check what you have learned. Drag the...
No. But, not right. But you notice what's happened now. We went from being implanted in mom to forming all these things, which are not just being a mom. So we have a name for that in biology land. So that's step number 3. The ugly word we use for that is gastrulation. Ugliest word in the universe. So in gastrulation, you form layers. So I went from a ball of cells to making layers, making structures. So the word for those layers are called the germ layers, or the embryonic germ layers. Can someone tell me from basic biology what germ means?
Grow.
Grow, germinate, right? So we're using the logic of a plant. So you're making these layers that will grow. We call that gastrulation. So let me show you a picture of this. Yes?
Is that within the blastocyte or around?
It is within and then also without. So it begins internally with the 2 layers, and then eventually the outside will move as well. So it's kind of both. Let me show you a picture of this gastrulation from another point of view. There we go, right here. So here we are burrowing into mom, week 1, right? So here you are, here's mom, bleh, just kind of eat your way in there, right? Do you remember biology 101 what troph meant? Autotroph, heterotroph?
Eat.
Eating. So literally, I'm going to eat you, right? Kind of thing. But then here we are. So here you are at 7.5 days, week 1. You've implanted. So pink is mom's uterus. This is you. So I'm chewing my way literally into her uterus. Here I am some more. But notice now we're getting layers forming. So instead of just a ball, I'm a ball with layers. So we call that gastrulating. So when we move ahead a little bit, notice I now have 3 layers, the yellow, the red, and the blue. These are not anatomically correct. You're never going to see that in the real world. But I have these layers and shapes within that ball. So you're still in mom's uterus, but now you're forming layers. That's gastrulation. Well, how old are you when you have layers? This is week number 2, so roughly 16 days. So step number 3, gastrulation, is week number 2. So 2 weeks following conception, you have gastrulated. Sounds gross, right? But you've gastrulated, which means you form these layers within that. So we're over here. You're supposed to know the name of the layers that you form, because thou shalt know them, thou shalt. Right? Luckily, there are only 3. So there are 3 germ layers, or embryonic germ layers. All right, here we go. Each one ends in the same suffix, which is derm. today? So all you have to know is the beginning words. So we have ectoderm, mesoderm, and endoderm. Medical terminology, what does derm mean? Skin or layer, right? So outer layer, middle layer, inner layer is basically how we're naming these. So if you look at my picture, if you look at the blue, the red, the yellow, look over here. These are my germ layers. There's an outer one in blue, a middle one in red, the inner one in yellow, to some biologist type. So we have 3 layers that form in the gastrulation event. Okay. So whoop-de-doo, what are the 3 layers going to do? They're going to differentiate. Good word. Into what? Those layers are going to make tissues. So the 3 layers are going to form different tissues in this implanted item. So we're going to list the tissues each derm creates, because they don't all make the same number and type. So to help you with that exercise, here's a picture showing my germ layers. Again, about 2 weeks along, 16 days. The blue and the red and the yellow are representing the different types. So if you look at the different layers, I have ectoderm in blue, mesoderm in red, yellow is the endoderm. If you read the little picture, they all make something different. So let's do each one. Let's start with the ectoderm, which is blue. What tissues does ectoderm make?
Nervous.
Nervous tissue is one of them. So if you go back to 231 and remember what nerves were, you got that. What other tissue does the blue one make?
Epithelium.
An epithelial tissue. A 231 word. So the ectoderm layer will make those 2 tissues out of it. All right. Mesoderm. Look at the red one. What tissues does it make? Connective and muscle. And some epithelial tissue. So it does muscles, connective tissues, and some epithelials in the blue. Look at the yellow, which is going to be our endoderm, the innermost layer. What's it going to make for me?
Epithelium.
Epithelial tissue. Again. Okay. So the 3 layers are differentiating into different tissues of the body. All right? So all 3 make some epithelial, 1 makes nervous, 1 makes connective and muscle. Why do I need tissues? What are tissues for?
Organs.
Organs. So the next thing I'm going to do once I have tissues is I'm going to start making me some organs. So after I make the tissues, I'm going to make the organs from that. And that's step number 4. Organogenesis. Medical terminology people, what does genesis mean?
Make.
Creation or make. So I'm making organs. So once I've made the layers, they make tissues and I can start making organs. How old are you, do you think, when you start making organs?
4 weeks.
Week number 3. So 3 weeks after conception, you're now going to start cranking out organs from these 3 layers of tissues. All right, so what organs do you think you're going to make? Let's see if we can figure it out. Can someone name an organ for me in 231 that had epithelial and maybe nerves in it? On the outside of you. What was it? Skin. So ectoderm will create the skin when I'm making organs. Can someone think of another organ that maybe might use one of those that begins with N?
Brain.
Brains are good. So your CNS will be from ectoderm, because your brain has nerves and epithelials, right? It's meninges. Right? Okay, hit me with mesoderm organs. If this were making organs, what organs might include these tissues?
Muscle. Heart.
So the heart is one. Some muscles are one. Right? How about bones? Say yes, bones. Bones are an organ, remember that? What comes out of your bones?
Blood.
Blood, yeah! Things like your kidneys, They have all that blood flow, etc. So these are organs that have muscles, connections, and usually some epithelials. Right? That leaves the endoderm. What organs would have epithelial alone inside of you?
Viscera?
Which part of your viscera?
Squishy part.
True. Be a little more specific, layer-wise.
Visceral layer.
How about the lining of your lungs?
Yep, that one.
The lining of my lungs, the inside lining, not the muscles or any of that, just the epithelial of my lungs. Name some other part inside of me that has epithelium besides my lungs. You studied in the first test. The intestines, the GI tract. Lining of GI tract. So not the muscle, the GI tract, or any of that, just the lining of that. How about your endocrine glands? Endocrine glands have a lining. They're inside of me. So once I make these 3 layers, they crank out these tissues, I start making these organs from those layers.
Well, like all the heart has nervous tissue and all
Yes,
that.
it does.
So would some of the ectoderm go to making that?
Yes, it has to migrate, but in terms of the big picture, it's basically your nervous system, the CNS.
Okay.
So let me show you this picture, not that one. Where's it at? There it is. Boom. So this is a diagram, figure, whatever you want to call it, showing exactly what I put here. Let's take a look at it. So we have the 3 layers of 3 germ tissues, blah, 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 ecto, endo, meso. And then over on the right, you have the organs that are made from that. So my ectoderm, things on the outside of me, like skin, hair, my brain and nerves, from the NT. Mesoderm is the middle of me, things like my heart, my bones, my muscles. And then endoderm is the lining on the inside of me, my lungs, my gut, my endocrine stuff. Because this is the more formal picture, so let's do this one. Zygote, time 0. Blastula, implant, week 1. Gastrula, week 2. Organs, week 3. So ectoderm is going to make my epidermis, my brain and nervous system. Endoderm is going to make the lining of my gut, the lining of my airway, the glands, things that just epithelial on the inside. Mesoderm does the middle. So muscle, bones, kidneys, dermis, blood vessels. So once I get to week 2, I can then make organs in week 3. Make sense? All right. So that's gastrulation. So you're going to do all that. Now I'm going to show you, where's it at? A little animation of that one. So we're going to kind of walk through exactly what we just did here.
During the first day following fertilization, the zygote begins mitotic cell divisions called cleavage
Yeah. Oh,
as
sorry.
it moves through the uterine tube. The cells formed are called blastomeres.
So
By
you see
the
that
4th
word
day
blast
after
all over the place.
fertilization, the blastomeres have formed a solid ball, now called a morula. By day 4 or 5, the morula enters the uterine cavity. Here, glycogen-rich secretions from the endometrial glands nourish the developing morula. The blastomeres in the morula are stimulated to rearrange themselves and form a fluid-filled hollow ball of cells known as the blastocyst.
That's the human word for it.
The cells continue to rearrange, forming an area of more cell layers called the inner cell mass. The blastocyst's outer layer of cells is called the trophoblast.
That's the eating part.
About 6 days after fertilization, the blastocyst attaches to the endometrium of the uterine wall.
So tell me, do you want your uterus thick or thin when all this is happening? That's the idea of thickening it. So before you ovulate, you thicken your uterus to get ready for that a week later. So you're going to have a really thick, bloody uterus because you want this guy to eat. Click the embryo. Right? Go. So now week 2.
During the second week after fertilization, the implanted blastocyst stage continues to develop. On the 8th day, the trophoblast of the blastocyst differentiates into two
So
layers.
this is week 2 we're doing. Okay,
The inner one is the cytotrophoblast layer.
Ugh.
The outermost layer, called the syncytiotrophoblast,
Say that twice.
secretes
All right.
enzymes that promote further implantation. In addition, human chorionic gonadotropin, a hormone necessary to maintain the nourishing secretions of the endometrial lining, is secreted.
We'll talk about that in a minute. So, don't worry.
Also on the 8th day, the cells of the inner cell mass differentiate into two layers. Together, the hypoblast and epiblast layers form the flat bilaminar embryonic disc. During the 9th day, the epiblast cells multiply and migrate, forming the fluid-filled amniotic cavity.
So now we're starting to break those into layers.
At the same time, the multiplying hypoblast cells begin to develop into a yolk sac. As a result, the bilaminar embryonic disc is positioned between the amnion and yolk sac. By the 12th day, differentiation of yolk sac cells forms an extraembryonic mesoderm. The migrating yolk sac cells, as well as the syncytiotrophoblast and cytotrophoblast layers, form the chorion. The chorion grows to surround the amnion and amniotic fluid and forms the connecting stalk, which is the future umbilicus. The chorion, destined to form the embryonic part of the placenta, is important in the exchange of nutrients between maternal blood and the embryo.
We'll do that next week. We'll do that part here. So now we've basically taken our implanted item. Now we're breaking the layers. So those layers are then going to start morphing into tissues and organs here.
During the 3rd week after fertilization, the 3 primary germ layers are formed, which provide cells for organ formation in the following months. These germ cell layers are formed by a process known as gastrulation, which involves rearranging epiblast cells. As cells from the epiblast migrate, a faint groove, called the primitive streak, forms on the surface of the embryonic disc. With the formation of the primitive streak, it is possible to recognize the head and tail ends of the embryo. Some epiblast cells migrate, differentiate, and then replace the hypoblast to form the endoderm germ layer.
So now we're getting a bottom layer.
Other epiblast cells migrate and differentiate to form a new layer between the epiblast and hypoblast, identified as the mesoderm germ layer.
Middle layer.
The remaining epiblast cells become the ectoderm germ layer. The result of gastrulation is the formation of the trilaminar embryonic disc, consisting of the 3 germ layers. The ectoderm gives rise to all nervous tissue and the epithelium of the skin. The mesoderm gives rise to blood, muscle, bone, and connective tissue. The endoderm forms into the epithelium of the gastrointestinal tract, the respiratory system, and many organs.
That's where we're at now. So now what we're going to do is we're going to start making this into actually formal organs. So to do that, we're going to go to this is a little website that Indiana University has that shows you how you can do that. So here we go. We're going to make organs from 3 layers. So when you're home, I want you to take 3 pancakes and then fold them into a person. So that's what we're going to do. So if you look at this, we have ectoderm here, and there's endoderm in here, and mesoderm. These are the 3 colors. So you have the ectoderm's blue, the mesoderm is red, and the endoderm is the yellow. So we have 3 layers from here. Now we got to form them into these over here, right? So how are you going to do that? Okay, so this is called gestational origami, right? So what would happen if I took the ectoderm and shoved it under that? Let's do that. So I pooch in the 2 sides of the ectoderm and bring the mesoderm back around over it, like this. Right? Notice there's now a space where your guts are going to go. That's your abdominal cavity. And then touch those together. Now you have a spinal cord that's off by itself. You have muscles around your gut, muscles around your intestines, and muscles under your skin. Now you can do that head to tail. This is really cool. I'm going to take this part and tuck it under. That's going to make a head. That's going to be mesoderm in here in your chest. That's called your heart. So I shove part of it down for a heart, part of it up for a head and a brain. This will be your butt here. This is going to be your belly button there. Cool, eh? So go home, get 3 pancakes, and make this. Right? So if you really want to do this accurately, you do them both at the same time, because remember, you're going to go 3 dimensions at once. So if I do this, so you're going to poke in the sides while you're flipping head to tail. So during your gastrulation event, you have to move the layers to each space that the organ needs to be. So what happened if your heart didn't end up there, but ended up over here? What do you think would happen to you? You're dead, right? Because for this to work, you have to put the organs in the right places at the right time. So you can try this. You can use, I failed that part of the class, but you can take clay and try to make a person out of it. I was really bad. Kindergarten was difficult for me, but that's the idea. So I'm going to take these 3 layers and then roll them into a person. Right? Yes?
So clearly that happens where the
Yes,
heart is
that's why we're
outside.
here.
So do you, I guess, so they just, it doesn't make it?
That's
It just- Okay.
You can, a few people can be born with things in the wrong place, usually not. A lot of people not very well. For this to work, gastrulation has to put every layer in the right spot.
So this is gastrulation, this is not organogenesis.
You've already got your forming organogenesis. So you gastrulate already, now you're forming the organs. So, by the way, there's a very famous quote, I don't know if you know this one, by a guy named Louis Wolpert. Gastrulation is the most important time of your life. Because if you don't have the right layers, you don't get the right you. Right? So this folding origami is kind of important for him. Right? But that's the idea that we're doing here. So by doing that gastrulation, we're then forming the right things at the right place. Right? So that leads us to some wording. So I'm going to show you some language here you might like, or maybe not. Where'd it go? I lost my paper, here it is. So, let's learn some language. So, fertilization, time 0. Sperm meets egg. 1 week later, you've done what? We've done at the end of week 1. Look on the screen over there. You what? Implanted, right? So, the old word for that was you were a conceptus. The newer word is pre-embryonic stage, but I don't care what you pick. But here you are, week 1, you've implanted. What happens at the end of week 2? You're gastrulated, so you're still a conceptus. Notice what you are week 3.
Oh.
You're an embryo. Why is week 3 a new name for you? You're making organs. So week 3, you've become an embryo because you're doing organogenesis. So week 3, organogenesis, you are an embryo by definition. So when you make organs, that makes you an embryo. So gastrulation, making layers of tissues. Week 3, you're now making parts, so we call you an embryo. All right? So now I'm an embryo because I'm beginning to make organs. So what do you think you are at week 4? Embryo? Yes? No?
I thought you said organogenesis was week 3, and that's
Yes.
when we make, that's
Yes.
when we become embryo.
Yes. So what do you think you're called in week 4? Embryo. What do you think you're called in week 5?
Embryo.
Embryo. What, week 6? Embryo. Week 7? Just
Embryo.
want there to be a fetus on here somewhere. Week 7?
Embryo.
Embryo. Week 8?
Embryo.
Embryo. Week 9?
Oh, fetus.
Boom. You changed your name. Okay? Why? What happened at week 9 that you're no longer an embryo? You have feet all the way back there.
You have a face.
No, that was way back actually here.
You look more.
Well, you look like a person who kind of, well.
Have you differentiated your gonads?
Okay, let's think about the logic. Why were you called an embryo? You're making organs. So I'm no longer called an embryo, therefore, I'm done making organs. Very good. So organogenesis is from week 3 to week 8. You're an embryo only during the time you're making organs. So therefore, let's put a big finale here on this one. Thank you. So, let's write that on the board. So week 3 to week 8, your embryo. Why again are you an embryo? You're making your organs, organogenesis. I'm forming parts. Week 9. Onward. You are a what?
Fetus.
Fetus. And why are you a fetus? Very good. You've completed major organs. Now, to be formal, you haven't built everything, right? You don't have genitals, but you would argue those probably aren't major. But you would, they are to you, right? But you have a heart, you have lungs, you have kidneys, you have livers, all the things it takes to keep you alive, you built here, right? So when you do that origami thing I'm telling you to do, finish it by week 9, right? And then you're done, yeah.
Are we still in step 4?
Yes, this is still step 4. We haven't actually gone past organogenesis.
So that origami type thing
Yeah.
starts in gastration or in
Actually, the folding starts actually in week. You make
Okay.
the layers here, but you fold them here.
Okay.
So to be honest, you make a person in 6 weeks, 3 to 9. Yeah.
So since organogenesis happens around like week 3 and 4
Uh-huh.
onward, is that why people have miscarriages and don't realize because they just
That's
think it's a period? It's
That's
at
right.
the same time period.
That's right. So according to most data, it's kind of somewhat self-reported. A lot of women are pregnant but don't realize it because you didn't get past basically week 2 or 3. So to you, it's just it's a menstrual cycle, right? So if this is true, why aren't you born at week 9? Wouldn't that make birth easy?
Yes.
2 inches long. What's wrong with being, if everything's built, why do you
You're
need to keep
dependent
going?
on the mother, you're a parasite.
Yeah, but why are you dependent on mom? I mean, if you're a kangaroo, you'd come out then, right?
You haven't completed the whole process yet. You've only done the major organ.
That's right. So what else would you need to do besides just build the organs? What's some other things you might need?
Organs need to work.
Organs need to work, so I need blood flow, I need arteries, I need all the enzymes. So it's going to take, even though you built the parts, you're not ready to move in yet, the logic goes, until you're further along. So you have lungs, but they're not big enough to breathe and keep you alive. You have skin, but you can't keep yourself warm, right? So even though you've built your parts, you can't live outside of mom yet. You don't have a digestive system that will handle food. The logic is you build a house, just because the roof's on, you can't move in. You could, right? So your major organs are done in week 9, hence the name change to fetus from embryo. You're done building big parts. So if you look at my cute little picture, right? You have feet, you have hands, you have eyes and ears, but you're not going to be talking to anybody today, right? They don't work in that level, right? Make sense? No, maybe you can, right? You can think in French, right?
You cannot, you know.
So if we take this another step further here, so here you are around week 4. So notice I've built organs, right? So I have a headish, footish, I have a cute little tail, little gills. There I am. Here I am at week 7. So I'm further along. But what am I at week 7? What is this? You are an embryo, because I'm still growing parts. I'm still building organs. Give me another week and I'm done with that, and then I can be a fetus. That's why the name changed to embryo in week 3 and then fetus in week 9. So that's kind of the logic of the name. Make sense? Okay, so now the question is, why didn't you menstruate way back in step 3? Remember you said last week on your test, right? You menstruate after you ovulate, which is 2 weeks later. So you should be menstruating right here. And why don't you menstruate here? How'd you get to here if you're supposed to menstruate there?
HCG.
HCG. Let's make a graph of some hormones. Not a graph, right? Okay. Let's just ask that question. Why don't you menstruate when pregnant? Let's ask that question bluntly. Right? The answer is hormones. The answer is always hormones. Okay, give me the hormones. That would be for the first 2 weeks there, week 1 and week 2. What hormone made where? It's on your exam.
Estrogen.
So you'd have the corpus luteum makes what? What hormone does the corpus luteum to maintain your endometrium? Estrogen and progesterone. Remind me from your test, what's the job of estrogen? To keep you thick, right? And progesterone's job is keep it bloody, right? Keep it maintained. So, as long as I have a corpus luteum, I should maintain my uterine lining. That would be from week 2, this week 1 and week 2. But now, the corpus luteum should go away, but it's not. So we're going to maintain the corpus luteum. We're going to make sure the corpus luteum does not go away. Doesn't decay. So if it doesn't decay, what will my endometrium do? It'll stay there, right? It's not going to menstruate. It's not going to go away. How can I keep my corpus luteum from going away? It was on the animation. They mentioned a hormone. The answer is HCG. So HCG is a hormone. I'm going to show you a graph. Boom. Like this here. So up there in, what was that, brown? Right? That's the name. You don't have to know it, but I'm going to write it anyway. Human chorionic gonadotropin. HCG is fine on a test. Right? But that's the hormone that maintains the corpus luteum. Its job is to keep your corpus luteum alive. If my corpus luteum is alive, my endometrium's alive. Therefore, you don't menstruate. Makes sense? Okay, so here's the question. Where does that come from? Told you in the animation a little bit. Where's it come from? From the chorion. Yeah, who has a chorion? Yeah, from right here, right? From the implanted embryo, from the implanted blastula. So, when are you implanted? Week what? Week 1. So from week 1 after conception, the blastula blastocyst begins to crank out this hormone. So if you look at that graph, one thing that's going to hit you is that's a doctor graph. The reason that's a doctor graph is because they number the weeks differently than biologists do. They measure from your last menstrual period, which is 2 weeks before you ovulate, right? So we do here. They don't. They're strange. But if I go from here, week 0, time 0, 1 week in, HCG is already climbing because I've implanted. Week 2, higher. Week 3, and forth. So once you implant, you begin to crank out HCG. Notice the progesterone and estrogen have never gone down. They keep going up. Right? So HCG's job is to maintain the corpus luteum. That way you don't menstruate, so baby gets a house. Make sense? All right. Let me show you that in another way. Where's that? Where'd it go? I think I lost it. Here we go. Boom. This will be a little bit of biology 112 in this one, but in case you ever wonder how peeing on a stick proves something, now you know. What do you think they're testing for in the pee? HCG, because the only time you should have HCG is when there's somebody living in your uterus, right? That's the logic of the system. So let's go through it.
A urine sample can be tested to determine whether a woman is pregnant. This test is based on the ability of antibodies placed on commercially prepared test strips to bind to specific molecules in the urine. The number of colored bands on a test strip determines whether the result is positive or negative. The urine of pregnant women contains
Can't
a
try that,
hormone
man.
called human chorionic gonadotropin, or HCG. HCG is produced by the developing placenta and has important functions in pregnancy. However, some of this hormone is also excreted in the woman's urine.
So stop and think about what we just took a test on, right? You have this floating in your body. You, as a woman, don't want it, so where do you put things you don't want? In your urine. So you're going to excrete this out, you're going to not reabsorb it, and therefore it's going to end up in your pee. So you pee on the stick, I'm seeing what's in your blood that you don't want, which is this. The baby wants it, you don't, right? So this is how that works. They're using the kidney function to show something in your uterus, basically.
Women who are not pregnant do not produce HCG. An HCG test strip can detect low levels of HCG in the urine. The strip is made of a capillary membrane that draws urine by capillary action along its length. The test strips contain 3 zones of function, here labeled R, T, and C, to indicate a reaction zone, test zone, and control zone. The reaction zone contains a type of antibody called anti-HCG antibody.
Okay, so this is 232. Remember, what do antibodies do? Please tell me you remember. I can change your grade for you. They stick to what? Antigens. So think about HCG is a hormone. It's a protein. It has a shape. You can make antibodies that stick to or against HCG. So they'll only stick to HCG if it's in your urine. That's the logic of that, using the immune
These are
system.
mouse monoclonal antibodies, which recognize a single molecular feature or epitope on HCG molecules. The letter E signifies that these mouse antibodies have been artificially linked to an enzyme. The test zone contains a polyclonal mixture of antibodies, which recognize a variety of epitopes on HCG molecules.
So these antibodies stick to dye and stick to those. So if these are working, you're going to get a color. So they're an antibody against the antibody, right? That's kind of cool.
This zone also contains dye molecules, which act as substrates for the enzymes currently in the reaction zone. The enzymes in the reaction zone can catalyze a color reaction with the dye substrate. The control zone contains the dye substrates, as well as a preparation of antibodies. These antibodies, called anti-mouse antibodies, can recognize epitopes on the mouse monoclonal antibodies that are currently in the reaction zone. The urine of a pregnant woman yields a positive test result. To determine this result, the test strip is dipped in the urine sample.
You pee on it.
The sample, which contains HCG, is drawn up the strip by capillary action and first arrives at the reaction zone, which contains the enzyme-linked anti-HCG antibodies. The enzyme-linked antibodies recognize parts of the HCG molecules and bind to these molecules from the urine. The complexes of HCG and antibodies, as well as the unbound antibodies, dissolve in the fluid and detach from the capillary membrane. They are then carried along the test strip by the capillary flow. The complexes and free antibodies arrive at the test zone. This zone contains the polyclonal antibodies and the dye substrate, both of which are permanently affixed to the strip. The immobilized polyclonal antibodies bind to HCG molecules in the complexes. The free antibodies continue to flow along the strip. The mouse monoclonal antibodies that escaped binding in the test zone have reached the control zone. Here, anti-mouse antibodies that are affixed to the strip bind to these monoclonal antibodies. In the test zone, the enzyme-linked antibodies catalyze a reaction with the dye substrate and trigger a color reaction. The same type of reaction occurs in the control zone.
So the logic is this one tells you that HCG is there. This one tells you that your urine went up to the strip. So 2 lines is a yes. If you were not pregnant, the HCG antibodies would not glow, but this one would prove your urine went through the test. So you get 1 line. The 2 lines is the dear we need to talk moment, right? And that's what they're doing, is they're checking for HCG using antibodies on the strip. Right? So EPT tests for HCG is what you're going to remember the rest of your life. All right.
Say that again.
EPT test for HCG. So if we go back to this graph here, last thing we're going to do. So when you start making HCG, roughly a week after ovulation fertilization, and it's going to climb as we get a bigger embryo throughout that first part of pregnancy. Right? So if you think about your embryo stage from weeks 3 to weeks 8, so I count 1, 2, 3, 4, 5, 6, 7, 8. So during the embryonic period is when I'm cranking out HCG to keep mom from dumping me, basically. So blastula is overriding the corpus luteum.
And why do we then not need HCG?
That's for next week. That's your placenta takes over. So once the placenta is there, this can go away because the placenta then will run the pregnancy out. Yes?
So if you take a pregnancy test after that period,
Over here?
yeah, so then would you,
Be negative if it's a relatively low
but
crosswalk.
you'd be pregnant?
Yes.
So they can say pregnant.
So the one the doctor would charge you $300 for would detect it. The one you get at the grocery store would say no, they're not that sensitive. So you can be 9 months along and show negative HCG, which means you're not pregnant. You just benign. So that's all for tonight.